The cost in drug research and development had been growing fairly steadily from 1950 to 2020. It has been estimated that a successfully approved new chemical entity or NME costs more than $2.5 billion and takes 13.5 years from discovery to the market. In the past decade, advances in many of the scientific, technological and managerial efforts have been applied to raise the efficiency of drug research and development. Scientists are seeking innovative technologies and approaches to reduce the cost and raise the efficiency of drug discovery and development. A novel drug discovery strategy, named drug repositioning, that identifies existing drugs for new uses, could deliver the promise of higher productivity. High-throughput technologies, such as next-generation sequencing, have generated a large volume of genetic and genomic data in several national and international cancer genome projects, such as the Cancer Genome Atlas and the International Cancer Genome Consortium. So far, these international or national cancer genome projects have identified hundreds of putative cancer driver genes that enable us to look at the cancer genome landscape. Moreover, the cancer genomics data have facilitated the revolution of a novel oncology drug discovery paradigm from candidate target or gene studies toward targeting clinically relevant driver alterations or molecular features for the development of precision cancer therapy. However, every day, millions of people are administering various anti-cancer medications that do not help them. In cancer, both somatically acquired and inherited mutations can mediate a patient's response to treatment. Recent advances in NGS technologies enable the identification of various genetic or epigenetic alterations. An urgent need is to develop more efficiently therapeutic agents that selectively target the particularly genetic profiles or molecular features. Precision cancer medicine needs to establish different types of clinical trials focusing on individual, not average, responses to therapy, finding the right drug with the right dose for the right person harboring a particularly genetic event or molecular feature. Time for one-person trials for precision medicine is underway. For example, U.S. National Cancer Institute launched the Molecular Analysis for Therapy Choice trial that will perform genetic screening for approximately 3,000 patients having advanced solid tumors and lymphomas to recruit 1,000 patients harboring particular mutations who will be eligible for one of 25 drugs, such as crizotinib and afatinib for non-small cell lung cancer with ALK rearrangement or EGFR activating mutations, respectively. In this course, we will systematically introduce the recent advances and development of individualized network-based drug repositioning approaches for oncology drug discovery and development by integrating large-scale cancer genomics data. Marriage of genomics data and systems biology enables development of the newly individualized network-based drug repositioning approaches or models for accelerating the development of precision oncology. We believe that the rational development and application of computational approaches and individualized network-based models in genomics era and a systems biology framework will motivate further studies on the application of big data in the oncology drug discovery and the development of precision cancer therapy and cancer care.